Hi, I'm John Toll, a student worker for Dr. Ostler in the biology department at Elmhurst University. According to the National Institutes of Health, over 500,000 people had leukemia in the United States in 2022. This year, an estimated 23,540 people have died from it. The good news, though, is that the death rate has been steadily declining since the 1990s, and that improvement comes from research. Our research is on the hematological impact of RUNX1 mutations. Hematological refers to blood and blood-forming tissues, and we're interested in mutations that lead to bone marrow disorders because bone marrow is where the blood is formed. These mutations cause disorders that can progress to leukemia, which is a cancer of blood and bone. RUNX1 is a gene involved in the formation of bone, fat, and cartilage, and it is one of the most mutated genes in cancer. The RUNX1 gene forms the RUNX1 protein, which is a transcription factor because it interacts with DNA. Our RUNX1 mutations are from leukemia patients, and the goal is to show that we can use our experiments as a test to determine if mutations are pathogenic, which means disease-causing. The hope is that this data will allow clinicians to better design treatment plans and ultimately improve their patient outcomes. To research on human diseases without using humans like you, we use human mesenchymal stromal cells, or MSCs. MSCs are derived from bone marrow. They're basically stem cells that can only become certain kinds of other cells. This process is called differentiation. MSCs can differentiate into precursor cells that then become myofibroblasts, adipocytes, osteoblasts, or chondrocytes, among other cell types. Those last three are fat, bone, and cartilage, respectively. The RUNX1 gene produces the RUNX1 protein, which I said before. It's known to inhibit MSCs from differentiating into preadipocytes, acting like a red light. When the RUNX1 gene is mutated in the MSCs, this often breaks the stopping function of the RUNX1 protein, so it acts more like a green light. We'll come back to this in a moment. Let's discuss that lightning bolt a little bit more. Lightning bolts are often used to represent mutations. Imagine that the DNA which is in this cell's nucleus is right at the tip of that lightning bolt. Remember that mutations change the RUNX1 gene, and that produces a modified RUNX1 protein. We'll come back to the protein after we discuss the cells a little more. The villager represents normal cells. The zombies represent mutated cells. So where did we even get these cells in the first place? We collaborated with Northwestern University's Godley lab. They created the mutant cell lines using CRISPR, and we conducted experiments in parallel with them. Our research involved two experiments. First, we compared cell growth over time. Second, we compared differentiation of those cells between the different lines. I'll show you the results of those experiments later. Let's talk about who's on Team Zombie. The zombie names and the shapes that I used are based on specific mutations. G69C has a different amino acid in position 69. This is a variant of unknown significance, or VUS. Now, for some of you, variants of unknown significance may sound similar to rodents of an unusual size, or ROUSs, but the two are not related. If you'd like to learn more about ROUSs, please watch the wonderful classic movie, The Princess Bride. Now, variants of unknown significance like G69C are important to research because they represent novel cancer patient mutations. So, discovering their features helps direct future therapeutic interventions for those patients. Now, D62VFS*9 has another different amino acid in position 62 instead, but the FS*9 is the important part because it indicates a frame shift in the DNA, which ruins the rest of the protein and truncates it. Truncations often lead to pathogenic mutants. Now, the last one, D62L67DEL, refers to a deletion of 6 amino acids in the middle of the protein, and we expected this to be pathogenic as well. Let's take a closer look at the RUNX1 protein. This is the RUNX1 protein, shown as a ribbon diagram. It links a protein above it to the DNA below. If you stretch out this ribbon, it forms a linear diagram. This is the villager version of the RUNX1 protein. The numbers indicate the positions of the amino acids in the protein, so there are 453 amino acids in the RUNX1 protein. The two most important parts of the RUNX1 protein are the RUNT homology domain and the transactivation domain. These domains are necessary for RUNX1 to bind to the DNA and the protein, respectively. So mutations in the RUNT domain cause major issues when it attempts to bind to DNA. Our zombies mutations are all in the runt domain. Green indicates the mutated area and black indicates the deleted area. And you can see how short the D62VFS*9 RUNX1 protein turns out to be, which is why I represented it just as a zombie's head. Our hypothesis was that the truncation and deletion zombies will grow faster and are more likely to become fat cells. We were not sure what to expect with G69C-VUS. Here's how the mutations impacted growth rates. We created this growth curve by counting cells every 3 to 4 days under a microscope for 21 days, while providing them with growth media, which is basically a nice warm bath full of food for the cells. As expected, the villagers have normal growth rates. The zombies, on the other hand, all exhibited much higher growth rates, including G69C-VUS, which was surprising to us since it was only a single amino acid change. The fact that the mutation was in the important runt domain is probably the cause. This leads us to classify the G69C mutation as pathogenic, just like the other two more severe mutations. The second part of our experiment was to induce adipogenic differentiation, convincing MSCs to turn into adipocytes, or fat cells. To do this, we added insulin and specific steroids to the normal growth media and gave this to wild-type and mutant cells, the villagers and the zombies. After 21 days, we stained them with oil red O to make the fats visible as the red blotches in the bottom photos. The amount of red is an indicator of how well the MSCs differentiated into adipocytes. We used a spectrometer to measure the change in the amount of red for each cell line. All the cells differentiated into adipocytes, but D62V frameshift zombies were obviously the winners of this fat contest. Interestingly, the VUS and deletion zombies were essentially the same as the villagers. They didn't produce more fat. We also had a negative control group, which we did not give steroids or insulin, although they were still stained at the end. This is what you see in the top photos, which show a lack of red blotches since the cells did not differentiate and become adipocytes. We confirmed our hypothesis that the zombies grow faster than the villagers, and the most severely mutated zombie formed more fat than the villagers. The G69C-VUS mutation was surprising because while it was a relatively small mutation, it grew much more aggressively than expected. Ultimately, testing different kinds of known RUNX1 mutations allows us to develop a reusable process with which we can test unique mutations from cancer patients to determine how those mutations impact cell growth and differentiation. You can think about it as, how bad is it going to be? It's important to understand the results of these mutations because they allow future experiments to focus on reversing those mutations with genetic therapy to restore normal RUNX1 gene and protein function, leading to viable clinical treatments for diseases like leukemia. We isolated RNA from each line of the induced cells. This includes mutated RUNX1 mRNA and all the other RNA currently active in the cells. In the fall, we'll use real-time polymerase chain reaction on the RNA to determine which of the RUNX1 targeted genes had changes in their expression. Eventually, we'll also sequence the RNA, although that may not happen this fall. Additionally, we plan to induce the same cell lines to become bone and cartilage cells instead of fat to determine what impact they have on those types of cells. If you're curious as to what that looks like, here's a diagram of how various transcription factors interact to convince MSCs to become various cell types. Start on the left side of the image with the MSC, and the arrows move outward with adipocytes at the top, chondrocytes at the bottom, osteocytes on the right. Now, there's a lot going on with this diagram, but you can see that the RUNX1 protein is involved in every one of the 3 different differentiation pathways shown here. So we expect our future experiments to be just as impactful as this one. Thank you to Priyanka Dixit at the Northwestern Godley Lab who provided us with the cells, and to my sons, Sam and Flynn, for listening to this presentation repeatedly. Sam also provided some Minecraft images for me. Thank you for listening.